So when you want to actually separate out, DNA or RNA or proteins, according to their molecular size, you use a technique that's called gel electrophoresis. And in gel electrophoresis, the molecules to be separated are pushed by an electrical field through a gel that contains small pores so that there's resistance to their travel. So the molecules will travel through the pores in the gel at a speed that is inversely related to their lengths, to how long they are. So a small, for example, negatively charged DNA molecule or a protein or an mRNA will travel a greater distance through that gel than a larger molecule, and it'll form these sort of bands, basically that will collect at specific distances. So what you do is you set up that gel, and you have a negative end, an anode, and a positive end, a cathode. And again, since amino acids, for example, have a net negative charge, they'll move towards the cathode, towards the the positive end. So this is a way to sort of measure the expression of proteins or the expression of mRNA by looking at the thickness or the intensity of these specific bands. And gel electrophoresis, again, current moves the proteins, and specific proteins form measurable bands in these gels. Now, Western blots are what analyze proteins. So a Western blot, is basically looking at proteins that are moving through the gel, from the anode to the positively charged cathode based upon their size. Not all proteins are fully negatively charged, or consistently so. So they are first treated with a detergent that actually unfolds them, and then coats them with negative charges, so they're sort of consistent in terms of the charge versus the length. But before we had a Western blot, there was something called a Southern blot, actually, which was developed by Edward Southern, who was or Edwin Southern, who was a British biologist. And in 1975, he published a method for detecting specific fragments of DNA, actually, after separating them by size. The DNA was first treated also with restriction enzymes that would cut and make specific fragment sizes, and then was added to this agarose gel, and then electrophoresis was carried out to separate these different fragments, DNA fragments by size. So Southern blots looked at DNA, and apparently they needed a lot of paper towels for the Southern blot process. They used stacks of paper towels to blot up the buffer when they were denaturing the DNA. So apparently after he published this work, the sales of paper towels to research laboratories just skyrocketed. And now ever since that, the Southern was his name. Western blot was just to say, it's another directional term. There's now Northern blots that look at mRNA, for example. There's an Eastern blot. So, there's a culture, of course, to science.